as usual, a couple of announcements this morning. First, thanks for coming, even though it was really slippery. Second one is on our right up here, literature review course. If you're looking for a 1-credit course next term, above and beyond the virology course and the mutant viruses from hell lab that you're taking, this might be really fun. Me and Steve Reichow in the chemistry department are going to be doing a 1-credit literature review seminar where we'll be discussing all kinds of primary literature on the most amazing viruses ever, i.e. the ones that we work with in my lab, but some other ones as well. And basically, this is really about thinking about methods, thinking about reading papers, analyzing them, etc. And there will be an essay at the end of the term. Otherwise, no other exams as far as that's concerned. If you're interested, please email me or Dr. Reichow about this. We will be giving preference to people who are enrolled in the mutant viruses from hell lab for all kinds of reasons that I'm not going to get into right now. So again, if you're interested in this, please let me know. Registration should be available as soon as you can register for classes for that. And again, it'll be instructor approval. So today we're going to finish up talking about translation. And if we get to it, talk a little bit about protein folding. A couple of people have asked me what's going to be on the exam on Friday. It will be everything through today's lecture. Wednesday will just be review on the stuff that, again, I think I did a crummy job of explaining the first time around. So last time we talked about, let me go back here, we talked about the genetic code, open reading frames, tRNAs, tRNA synthetases, and this will be the aminoacyl tRNA synthetases, and then the ribosome, which is this absolutely amazing structure, and we now know literally in atomic detail how the ribosome works. And then today we'll talk about how you start translation, a little bit how translation elongates. Again, this is the alphabet soup part here. Lots of different initiation factors, elongation factors, release factors, and then some quality control, protein quality control, messenger RNA quality control, and then if we get to it, talking about proteins. So again, it's really all about this last step right here. We're going from RNA to protein and how you actually finally get that protein at the end. So let's start out with the bacterial system. This is the more straightforward system in terms of number of proteins involved, et cetera, but it's exactly the same problem and issues that eukaryotic translation has to deal with as well, because these have been completely conserved all the way through cellular life. So the main things that have to happen right at initiation for translation are going to be getting that initiator tRNA to the start codon, which is in the middle of the ribosome, in the P site, but also making sure that you don't have the large subunit of the ribosome coming in and binding to the small subunit of the ribosome, because if it does that, there's no way you can get that first tRNA into the ribosome in the first place. So the last thing that has to happen is you want to make sure it's happening just at the P site, right in the middle, as opposed to at the A site, which is normally where you're going to be having tRNAs associating. We'll take a look at that in just a second. So we have these IFs. What the heck is an IF? An IF is an initiation factor. So IF, and again, 1, 2, and 3, great, wonderful names. The really important one here is the one in the middle, IF2. IF2, and we'll see this also for the eukaryotic initiation factor 2, that's the one which is associated with the initiator tRNA in the P site in the ribosome. IF2 is a GTPase. This should sound very familiar because we talked about GTPases in terms of timing molecules in the past. GTPases bind to GTP. Then there's hydrolysis of that GTP to GDP, which causes a structural change in the protein. And so that's what's going to be happening here, IF2. And then once that happens, you can have the association of the rest of the ribosome. IF3 is blocking the large subunit from binding. IF1 is blocking tRNAs from binding at the A site. So these are the 3 initiation factors that you need for bacterial translational initiation. IF2, holding on to the initiator tRNA, making sure that you have the right base pairing interactions. Then we have GTPase activity, that's released. IF3 blocking the large subunit of the ribosome, IF1 blocking binding by tRNAs in the A site. So that's what happens in bacteria. Again, appropriate association. How does the ribosome associate with the tRNA in bacteria? Or sorry, not the tRNA, the messenger RNA appropriately? Shine-Dalgarno sequence, exactly. So complementary base pairing interactions that happen in bacterial ribosomes. It's not true in eukaryotic ribosomes. So we're shifting over to talk about eukaryotes now. Eukaryotic translation initiation uses AUGs, as we have here in the middle, but it's not just the AUG, which is important, not just the start codon. If you do comparisons of sequences, and I don't think we've looked at this, but hopefully Dr. Bartlett talked about these what we call sequence logos before. All that the size of each of these letters represent is how frequently you have this particular nucleotide at this particular position. So 100% of the times you have A, U, and G at these three positions, but most of the time you're going to have a purine here at this position, an A or a G. Most of the time you're going to actually have a guanine at this position. Most of the time you're going to have a cytosine at this position. What this means is that the ribosome is going to preferentially start here, but not always start here. And so this is unlike the case with the ribosome binding site that you have in bacteria. There's always that specific, very specific individual nucleotide binding. Here it's a little bit more flexible. And that's because the messenger RNAs in eukaryotes don't have these Shine-Dalgarno sequences. They don't have the ribosome that binds in the middle of an RNA. All of the initiation complexes happen at one end of your messenger RNA, and that's going to be at the 5' end. I do not expect you to remember the vast majority of things on this slide. Basically wanted to point this out as why we should care, other than the fact that there's going to be an exam on it on Friday. This is about cancer and cancer biology. Turns out that many of these initiation proteins, eukaryotic initiation factor, EIFs, are misregulated in many different cancers. So it's not just, Stedman's forcing us to memorize these things. But it turns out that any kinds of changes in these translation proteins do turn out to be really important for cancers, which is kind of strange because so far all we've talked about with cancers is genetic disease and mutations, which are important. But it turns out that it's expression of a lot of proteins. When I say expression, it's the making of those individual proteins that can also be really important in terms of cancer progression. So let's get to the level that I expect you to remember this rather than the people who are talking about these cancer things, is what's going on in eukaryotic initiation. And this may seem just completely overwhelming. It was to me the first time I looked at it. But basically, think about this from the point of view of what we just talked about right at the beginning in terms of bacteria. So IF2, initiator tRNA, IF1, blocking the A site, IF3, blocking the large subunit association. So here, all you need to do is put an E in front of it. Eukaryotic initiation factor 3 does exactly the same as IF3. Eukaryotic initiation factor 1A does the same thing as IF1. Eukaryotic initiation factor 2 does the same thing as IF2. So these are really easy to remember. The difficulty and confusion comes about because we've got all this funky changes that are happening at the 5' end of our messenger RNA. We've got a cap, we've got poly A tails, etc. So these are really different between the bacteria, and the eukaryotic system. So what's going on here at the ends? We have EIF4E, which binds to the cap, and EIF4G, which forms kind of a looping structure. It interacts with the cap and with the poly A tail. So anytime you're thinking about a eukaryotic messenger RNA inside the cytoplasm, it's going to have this looped structure, which is one of the reasons why that animation we looked at last time is actually wrong, because these structures are always looped around next to each other, and that's because the 5' and the 3' end are associated due to this eukaryotic initiation factor 4G. And that's probably helping to both stabilize the RNA, but also helping to get multiple rounds of translation to take place. I think one of you had a question about that last time. Do you have multiple ribosomes associated with any given messenger RNA? Yes, definitely, and very much so this case, and that's really helped by the fact that the 5' and 3' ends are right next to each other. So how do you get these things associated with the ends of the DNA? Remember we had our cap binding complex, right? That's what comes out with the messenger RNA when it's exported from the nucleus. That's then just exchanged for AF4G. But then how do we get the small subunit of the ribosome associated with this? It's not associating because, again, of a Shine-Dalgarno sequence. It's associating based on protein-protein interactions where we now have all of these EIF4E, EIF4G, now associated with AF2 and the small subunit of the ribosome. This is all not the 5' end of your messenger RNA. The AUG is not the 5' end of the messenger RNA. So somehow this ribosome has to, say the small subunit of the ribosome, needs to move its way along to get to this start site. And that's the activity of yet more replication, translational initiation proteins. This is ATP hydrolysis, which allows the small subunit of the ribosome to move along the RNA, searching for the first AUG, but also getting rid of any secondary structures that happen to be in that RNA. And so we talked about secondary structures you have in RNA. Secondary structures, just two strands that are associated with each other. They're associated with each other. It's a helix, so you have to have the helicase activity breaking those things apart. So you have helicase activity. Why do we talk about helicase activity in spliceosomes? Because you need lots of ATP hydrolysis. You need exactly the same thing here for getting the small subunit of the ribosome to move along to the AUG. Once it gets to this AUG, now you have base pairing that forms between the initiator tRNA and the start codon, eukaryotic initiation factor 2, which was bound to GTP. What happens to GTP? Gets hydrolyzed, and then EIF2 is released, EIF1 and EIF3 as well. Now you have the large subunit of the ribosome, which will come down and bind. And here it's shown as coming on top, but as we saw last time, it's really coming in and sort of forming a sandwich between the two because it's hiding those RNA binding sites, tRNA binding sites, right in the middle. And now you're basically ready to undergo translation. You've got a tRNA in the P site that has an amino acid in it, just waiting for the tRNA to bind to the A site and have this transpeptidation take place. So we have the next amino acyl tRNA binds, then, transpeptidation takes place. Again, all of this is catalyzed by the large subunit of the ribosome. So now we have two amino acids attached to the tRNA, which is now in the A site. What has to happen next? Everything needs to move, exactly. So that movement takes place through the ribosome, and it turns out each different piece of the ribosome, the small subunit seems to move before the large subunit moves. And this is a process called translocation. So this translocation, the first thing that happens is it depends on your point of view, but you've got the small subunit, which is down here, the large subunit, particularly with that A site, now our tRNA with the extending peptide chain in it is in the P site. Now we just need the small subunit to move over. Now we're ready to go and can cycle back, do this whole process again. This does work in the absence of any other proteins, but it's really, really slow. So there are now, the next step, we had our IFs. What comes after I? E, elongation factors. But first, yes, question.
Sorry, so the small subunit moves before the large subunit, and that just shifts their position?
Well, these are all relative, so they're going to be moving independently of each other. So you can either say, oh, it's the large subunit which is moving this way, or the small subunit which is moving that way. It's all relative. So, but they don't move at the same time, and that's the important thing here. Yeah?
Is that a proofreading factor?
Is this in terms of proofreading? No, this has nothing to do with proofreading. It's just a translocation event. So the proofreading, which actually has more to do with fidelity, we'll get to in just a second. So it's all about how you get that appropriate amino acid in that A site, or I should say the aminoacyl tRNA. So how do you help this? It does work, but again, it's incredibly slow in the absence of extra proteins. So now we need the EFs. We had the IFs already. Now we need the EFs. EF is just elongation factor as opposed to initiation factor. And as we've had before, the bacterial one has nothing in front of your EF, and the eukaryote one is the eukaryotic elongation factor. Unfortunately, they don't have the same names, EFG, elongation factor G, or eukaryotic elongation factor 2. But they do exactly the same thing. Both of them are GTPases. EFG, or eukaryotic initiation factor 2, will bind to a ribosome where you have a site tRNA with this peptide chain on it, and then basically either pull the large subunit or push the small subunit. Again, this is all relative, in such a way that now EFG, or eukaryotic elongation factor 2, is sitting in the A site together with GTP. What happens with the GTP? It gets hydrolyzed. You have a conformational change here, which basically kicks the whole ribosome along. Now you have a free A site, just like you had up here at the beginning, and you can continue to go through this whole process. So elongation factor G is not absolutely required, but it really speeds up translation because it's helping the ribosome to move along. The other one of the elongation factors is elongation factor 1 in eukaryotes, or EFTU in bacteria. And in fact, we looked at the EFTU structure way back when we talked about G proteins originally. It was exactly that EFTU structure that we were looking at that changes when you have GTP hydrolysis. So EFTU binds to tRNAs, and this is one of the other things in that video that we looked at last time. You had all those little tRNAs floating around with the little red dots at the end, those amino acids. All of those in the cell are associated with EFTU, or eukaryotic elongation factor 1. So they're not freely floating around. They're all bound with this elongation factor. So this elongation factor, together with the tRNA, will bind to the A site. And if this is the correct base pair interaction, then you have GTP hydrolysis. That amino acid is released. Now you can have transpeptidation, etc. However, if you don't have the right interaction here, and I really dislike this proofreading here. I like to just call this fidelity, because it's if you have the incorrect amino acid that doesn't base pair correctly here, the EFTU and the tRNA will dissociate. So it's bouncing on and off all the time. And if you think about it, there are 20 different amino acids, and depending on the number of tRNAs, you've got 30-odd in a minimum case, as many as 40-some in a human cell. You're going to have a lot of incorrect interactions that are happening here. And so that whole process, and they do show this in that animation we looked at last time quite well. We've got the tRNAs coming in and coming out, coming in, coming out, and then only occasionally having the correct one which comes in. And because of this, translation is actually really pretty slow. Compared to replication, compared to transcription, translation is really, really slow. Almost 10 times slower than any of those other mechanisms that we've talked about already. And a lot of that is because you're checking to make sure that you have the right interactions that are going on here. Clicker question already? Gave people a bit lower time to get here. Everyone got their clickers out? Can we start yet? No? Wait. Give us a second, please. I'll read it first. What seems to be the main role for ATP hydrolysis and translation initiation by eukaryotic ribosomes? Ribosome translation from codon to codon, removal of secondary structure messenger RNA, checking to make sure codon-anticodon pairing is correct, coupling to transpeptidation, or association with a large and small subunits of the ribosome. You're awfully quiet. Ten, 5, still a big bump here at the end. We're still going for 100%, right? Yes? Are we going to get 100%? It doesn't look like we're at 100% on this one. So ribosome translation from codon to codon depends on what kind of hydrolysis? GTP hydrolysis, not ATP hydrolysis. Removal of secondary structure messenger RNA, that's due to what? Helicases, RNA helicases. It's pulling those two things apart. What do RNA helicases use? ATP. And codon-anticoding pairing is correct? GTP. Coupling to transpeptidation. That is important. You do need ATP for that, but where does that come from? Amino acyl tyrosine synthetase. Yes, I know that was last week. So an association of large and small subunit of the ribosome, you definitely don't need any kind of hydrolysis for that. So what is our answer? B. Okay, so that brings us to what I thought we were going to have for our next slide, which was this one, which is a really amazing movie that was done looking at where EFTU is relative to the ribosome and how it checks to make sure that this proper thing is happening. But before that, I'll try and answer your question.
Why is it that GTP is hydrolyzed in some settings and ATP in others? Is there like a symbol according to that, or is it just kind of like mechanically is what it is?
So basically, I'll paraphrase. Why GTP and not ATP in these kinds of situations? And the answer is I don't know. I know there's some theories about why, but I think it mostly has to do with the binding proteins themselves. And that binding seems to be more specific for G rather than A, and it's probably because ATP is used in so many different places, you're mostly using GTP for these extra structural changes which are happening. But that's a lot of hand waving at this point. It is what it is, and we can wave hands on why that is. Yeah?
The fourth slide, it said that once mRNA binds to the small subunit, ATP is used to find the AUG. So what is that?
So the ATP is used to find the AUG. That's moving the small subunit of the ribosome along and getting rid of any kind of secondary structure which is in that RNA. So I didn't have any movement from codon to codon in that place. And if that had been one of the possible answers, translocation of the small subunit of the ribosome to the ATG, that also would have been one of the things that ATP is being used for. But that wasn't one of the selections that I had in. So come up with these questions. What did I spend all weekend doing? Writing exam questions. So is it done yet? No, because I need to know what we're talking about today. So I did want to show this, as I say, really amazing video. Mostly what this is about is looking at the ribosome, where we've got, sorry, it's a little hard to see the arrow here, but there's the tRNA in the E site in yellow, the tRNA in the P site in green, and now what we're going to look at is where the tRNA in the A site is coming in together with EFTU. And then at the bottom is the gray. Here's EFTU with that amino acyl tRNA. Interacts with one of the proteins, interestingly enough, in the ribosome. That's that brown thing that's moving there. And now this is where EFTU in the red and the amino acyl tRNA in the purple is interacting and basically checking to see if it's the appropriate base pair interaction, which is forming between the codon and the anticodon. So here, and again, we're just zooming in and taking a look at that. Now this is part of the bottom there in the, I guess what color is that? Cyan, I guess. Small subunit of the ribosome is now actually interacting with pieces of that tRNA which are not at the anticodon. This is now at the amino acid end. If you have now the appropriate interactions with the codon-anticodon, now you have hydrolysis, and here's this hydrolysis taking place of that GTP. That then causes a change in the structure of the red EFTU protein, which will now release the tRNA, which will swing over to where you have the transpeptidation site. The empty tRNA that doesn't have an amino acid on it anymore is released, and the whole structure will repeat here. I'm still blown away at how much we know about the structures of all of these things. But it's about that, the interaction that's happening between the codon-anticodon loop that gets transferred to the part of EFTU that's going to hydrolyze that GTP through the small subunit of the ribosome, and then the appropriate interactions causes GTP hydrolysis. Those lack of appropriate interactions, which is not shown here, then EFTU and that tRNA is going to come off of the ribosome, and the next one will have to come into it to move through there. There's a, excuse me, a nice description of this, the whole paper, crystal structure of the ribosome bound to EFTU and amino acyl tRNA. This was, wow, 2009, so already 9 years ago now. Again, the structures of the ribosome now are really been really incredibly well understood. So when I've talked about initiation, we've talked about elongation. Now, last part, termination. So termination of transcription happens when we have the ribosome translocates, thanks to EFG, and now instead of having a normal codon in that A site, now we have one of the stop codons. Well, there's no tRNA that can associate with that. So what associates with that codon in the A site when you get to a stop codon? It would be really nice if they actually had the TFs, the termination factors here, but no, they came up with different names for them. They just called them release factors instead. So release factors bind to the ribosome in the A site where there's a stop codon. And basically what this does is it mimics a tRNA, only that doesn't have an amino acid up here at the end to have this transpeptidation reaction take place. So transpeptidation still takes place, but now it's to water. And so instead of forming a peptide bond between the COH, which is the end of your chain, and whatever the next amino group it is, it now will just hydrolyze that bond and the peptide is released. Once you have the peptide that's released, the ribosome falls apart. Okay, the ribosome falling apart here. This is basically the way our textbook looks at it. Again, this textbook was from, ages and ages and ages ago, so we now know a lot more about what's happening in terms of this particular, really this last step here. So what's going on? Again, with a much higher resolution structure now. If you look at the release factors, these release factors really do look like, and we'll see an X-ray crystal structure, an atomic resolution structure of one of these in just a second here. These really do look like tRNAs. It may not look quite like one here. And then they all have these glycine, glycine, glutamine here at the end of the protein, which is mimicking, again, where we would have normally your amino-acyl tRNA that will get attached to the growing chain, which is there. So, oh, hang on, we got our clicker thing on top of this here. It might be useful to move it away. So first step, and the first step up here is basically what we looked at already. We have our release factor. That release factor associates with the stop codon and causes this transpeptidation to happen. The problem is that now we have a ribosome that's got a tRNA in the P site, a tRNA in the E site, and a release factor in the A site. So how do we get this ribosome disassociated so it can reassociate again and start? I give you one guess. What happens in all of translation? GTP hydrolysis, exactly. So I didn't mention this before, but this is RF1, release factor 1, that associates here in the A site. But then we have RF3 bound to GDP, which now hydrolyzes. That hydrolysis will allow the release of the release factor, but we still have the ribosome with a tRNA in the E site and the P site. So what do we need? More GTP hydrolysis. But now it's the elongation factor G, which would normally be binding to the ribosome. Again, once you've had transpeptidation that takes place, you're going to be moving the ribosome along. Well, now it moves the ribosome along in such a way that the whole ribosome falls apart because you don't have that peptide bond still associated with your tRNA, which is sitting in the P site. So we have EFG, which hydrolyzes GTP. The whole process falls apart. And just to be sure that you don't have reassociation, the first thing that interacts now is IF3. What the heck was IF3? IF3 was that initiation factor, which is blocking the large subunit from associating. So the important thing is we've got a small subunit which is now free and available to go and find a new ribosome binding site or 5' end of your messenger RNA in order to restart this cycle over and over again. So binding to the site, the A site, by first release factor. Then you have a GTPase with that next release factor, which will remove that release factor. Then EFG, or eukaryotic elongation factor 2, which will allow you to move the ribosome along. That movement of the ribosome in the absence of having a peptide associated with it pulls the ribosome apart, and so you can start again. I mentioned that I would show you this before. Here's a tRNA structure, and this is the X-ray crystal structure of one of those release factors. I believe this one is release factor 1. I'm not absolutely certain. But this alpha helical structure here is what's also called the peptide anticodon, because it's amino acids that are basically interacting in a very similar way to the way that the codon and anticodons would be associated at this end of the tRNA, and that conserved GGQ residue at the end of your protein corresponds to where you would have the amino acid in a charged tRNA. So that's the whole process: initiation, elongation, termination. Let's look at more of how these things are fitting together. And I mentioned the loop structure already, multiple ribosomes associated with one messenger RNA. Why is this important? We went way back to the beginning of the class. We talked about transcription as being a way to amplify the product of one gene. Translation is also a way to be amplifying the product of a gene. So if you have one gene and you want to make a whole bunch of protein from it, you want to have a lot of transcription and a lot of translation. I also mentioned that translation's really slow because you've got to screen through all these 20 amino acids to figure out that you've got the right one. And so in order to get a lot of protein from one messenger RNA, you need a bunch of ribosomes on that messenger RNA. And this is, in fact, what you see if you isolate translating ribosomes from a eukaryotic cell. You see this thing called a polysome. And it's just literally every single one of these blobs here is a ribosome. The messenger RNA is this sort of bright streak that you have along the bottom here. And as I mentioned before, cap binding protein, initiation factor 4E, associated with the EF4G, which then binds to the poly A binding protein, which is associated with the poly A tail. So you have translation starts here, ATPase gets you the first AUG, elongation all the way around this messenger RNA, you get to the stop codon, the ribosome dissociates, but it's right next to where it can reassociate and start again. So if you think about how messenger RNAs are being translated, in eukaryotic systems you always have these polysomes. In bacterial systems, since translation and transcription are happening in a coupled manner, as soon as you have the ribosome binding site which is made, the ribosome will be binding onto that and then continuing. You also will have recycling of all these ribosomes. It's not going to be as fast as you have with a polysome because the 3' end is not right next to the 5' end. But if you think about a polycistronic messenger RNA, you'll have the ribosome binding site for one cistron, one protein is made, the stop codon will be there, then the ribosome comes apart, the next ribosome binding site is right there. And so you can continue along in that process. So this is what these eukaryotic polysomes look like. And again, you've got, for the bacterial ones, all of these ribosomes, but also lined up along one messenger RNA. So now I want to talk about a couple of exceptions to how normal translation is going to work. And these have to do with one really cool virus description, but also the so-called 21st amino acid. So we looked at the genetic code already. Yep, 20 amino acids, great. 64 codons, 3 of those are stop codons, 1 start codon. But people found when they were looking at proteins that there were, so it turns out that there are 3 of these really bizarre ones, but we're just going to talk about one. Selenocysteine. So selenocysteine, cysteine normally has an SH which is attached to it, which is great for forming disulfide bonds to stabilize proteins. But in some proteins, people found that there was this selenium that was sitting in a protein. And people spent a long time trying to figure out how on earth that cysteine actually, the selenium got into any of these proteins. Some of these proteins are absolutely critical for function. If you don't have enough selenium in your diet, you'll die because these enzymes are absolutely critical for a number of different processes inside the cell. Fortunately, you need very, very little selenium. But you do still need a certain amount of it. And then how the selenium comes in is actually a really fascinating process. It turns out that there's a specific tRNA for selenocysteine that has the anticodon UCA, which, as all of you remember from the genetic code, I wouldn't remember myself, is going to bind to UGA, which is one of the stop codons. So now you have an tRNA which can associate with one of the stop codons. That then has a serine aminoacyl tRNA synthetase that puts a serine onto it, and that gets converted to selenocysteine. Now selenocysteine on this tRNA that normally would be binding to a stop codon associates with a protein which is very similar to EFTU. Now, if you had this, and you do, all of our cells have this selenocysteine tRNA with selenocysteine associated with it, that's going to base pair with a stop codon. if all of the stop codons that we had in our genomes associated with selenocysteine, it would be a mess. And selenium is a pretty active metal ion. So you wouldn't want to have selenocysteine attached to the end of all of your proteins. You only want to have it associated under very specific conditions where you actually happen to need that particular amino acid. So how does that happen? It happens because there's a secondary structure in your RNA, and this is just one version. There are 3 different ways that this can happen. But this one is you have a secondary structure that forms in the messenger RNA 3' to this normal stop codon, which associates with this elongation factor that brings in the selenocysteine tRNA. This then binds perfectly happily. The ribosome has no clue what amino acid is here. That gets bound. You have translocation, etc. Everything is happy here. So this process is called recoding. Why is it called recoding? Because normally, what does UGA stand for? It's a stop codon. So normally it would be a stop. In the presence of some extra structures and extra proteins, now the ribosome gets a little confused, and the main confusion, again, has to do with this specific tRNA, which binds here. But it's only going to bind here if you have this extra secondary structure, which is present in the RNA. And this is how we get selenocysteine incorporation. Yes?
So how does this affect termination? Does it stop there, or does it try to keep going?
OK, so the question is, how does this affect termination? Because you've gotten to a stop codon, what happens next? Well, what is the next codon? Is the next codon a stop codon? Not necessarily. And in fact, most of the time it isn't. And the ribosome is just going to keep chunking along. You're going to have EFG. Now, at this point, once you have transpeptidation that takes place here, as far as the ribosome is concerned, this looks like a perfectly normal tRNA. This looks like a perfectly normal tRNA. The elongation factors are going to move the ribosome along. Whatever's the next codon is going to be incorporated, et cetera. So usually these selenocysteines, not surprisingly, are going to be in the active site of a particular protein. And those active sites are usually not right at the end of the protein, usually quite in the middle of the protein. Yeah, sorry.
So that loop is a sequence of mRNA associated with
So yeah, so just again, to paraphrase your question here, it says that sequence, and so actually it's more of a structure which forms in your messenger RNA, then brings in the elongation factor that's associated with the selenocystyl tRNA. If, as we're getting back to the question here earlier, if that's not the end of your protein, yes, those are also going to contain codons, which are in there, which will be complementary to each other, because otherwise you wouldn't be forming this stem loop structure. Other questions on selenocysteine? So another really fascinating aspect of changing translation also has to do with these secondary structures which are going to form in the messenger RNA. In mostly retroviral RNAs, we have these secondary structures which form, and in this case it's just shown as being a hairpin loop structure. This hairpin loop structure here basically changes where the translocation of the ribosome happens relative to the messenger RNA. So what happens in HIV and again a number of other retroviruses is that most of the time the ribosome will translate this protein, but occasionally, and it's at about 10% of the time in most of these retroviruses, the presence of this loop, which is present in both cases, so 90% of the time the ribosome just keeps going through here, all the codons are here as you were mentioning, gets translated normally, gets to a stop codon and finishes. In 10% of the cases, the ribosome doesn't translocate properly. And this usually happens where you've got a whole stretch of nucleotides that are exactly the same. So here, for instance, this stretch of 5 U's here. So instead of moving 3 nucleotides, it only moves 2 nucleotides. And in that process, you end up with a different amino acid here, but you've also changed the open reading frame. And so from last time we talked about open reading frames, again, that's where you have started, because you're going to go 3, 1, 2, 3, 4, 5, 6, 7, 8, 9. But if you have a frame shift, that's going to move that 1 nucleotide over and give you a completely different protein sequence. And in the case of these viruses, they give a fusion protein which is much, much longer, because the stop codon, which is here, is no longer recognized as a stop codon and makes a much longer protein here. Why is this useful for the viruses? It's because most viruses have relatively compact genomes, and this allows those viruses to use much more of the information which is present in that genome and make different proteins from it. So I think when we talked about open reading frames, only one open reading frame is used. You remember that quote that I put on that slide? This is a really nice example of how multiple open reading frames are actually being used in usually viruses. There are a few cellular cases, but the vast majority of these are going to be viruses as well. So now I want to shift gears a little bit and talk about quality control. Fidelity would be another way of thinking about it. But this gets back to the fact that ribosomes are really slow. You need huge amounts of nucleotide triphosphate hydrolysis. GTPs, as we've gone through now probably ad nauseum, GTP hydrolysis for translocation, GTP hydrolysis for checking for the fidelity to make sure that you've got the right amino acyl tRNA synthetase in any given position. But also lots of ATP, not so much for the very beginning in your initiation, but for charging all of those tRNAs. Charging the tRNAs, you need at least 2 ATPs. For any elongation step, you need at least 2 GTPs, which are being hydrolyzed. So that's 4 NTPs for any single amino acid. How many NTPs do you need to hydrolyze per individual nucleotide that's added? 1. How many nucleotides per codon? 3. So now we need 4 per any amino acid which is being added. So translation is actually the most energy-intensive process of replication, translation, replication, transcription, translation, of anything which is happening in the cell. So translation is really where all of the energy is going in terms of what's happening inside the cell. Now, there's other prospects as well, but in terms of molecular biology, it's about the translation. So what that means is, if you have problems in translation, you want to fix those problems, and you want to fix them quick. You want to have lots of ways of dealing with that. One of the really major problems that happens with all nucleotides is those nasty nucleases, right? They love to break your strands. You've also got hydrolysis that loves to break phosphatidester bonds. So broken messenger RNA is a big problem. Now, how is that dealt with? In many cases, one of the ways you can deal with a broken messenger RNA, or certainly protect it from the exonucleases, you circularize it, so the 5' and 3' ends are associated with each other. And then there's this neat protein and RNA process in bacteria called tmRNA, or SSRA. So what does this do? This is actually, for a change, reasonably well named. tmRNA is a combination of a tRNA and a messenger RNA. So tmRNA, which is also called SSRA, this is the gene for this, but I call them tmRNAs. This tmRNA has a part of the RNA which looks just like a tRNA and actually gets an alanine associated with it. This can associate with the A site of a ribosome if there's no messenger RNA in that A site. So this is what would happen if you have a broken messenger RNA. There's been endonuclease activity, exonuclease activity, which just chewed into it before it gets to a stop codon. So now the tmRNA can associate here at the A site. This part of your tmRNA looks like a tRNA, but the rest of the RNA here is acting like a messenger RNA. So you have transpeptidation that takes place right here. The protein is now attached to this tmRNA, and now this mRNA part is sitting in the ribosome in the A site. You can have normal association with your aminoacyl tRNAs here and extension of the protein all the way out to the end here. Now what does this do? It gives us all of our orange amino acids here. These orange amino acids are those that are coded for by this tmRNA. Now if you had a protein that was made by a broken messenger RNA, is that likely to be a good protein? No, highly unlikely to be a good protein. So what does this 11 amino acid tag say? Eat me, exactly, to get rid of this protein. This is clearly a bad protein. And that's because the eat me encoding sequence is right here on the mRNA part of the tmRNA. Yeah?
So does the ribosome signal that it needs the tmRNA because it's reached a broken end? Or what signals the tmRNA?
Right. So the question is what signals the tmRNA to come in? Basically what happens here is that this ribosome is stuck. It's not moving anymore. And if you have a stalled ribosome, that's the signal which says, okay, it's time to get this tmRNA to come in and do that. So it's the case of having a stalled translational machinery.
So if it's going to degrade it anyway, why does the protein not just go in and dissociate the ribosome and just say, okay, never mind?
So I guess maybe I'm missing your question here. So why does the ribosome go to this? Well, say the whole machinery, go through this whole machinery in order to get rid of this particular protein and whatever's made here.
And makes it incomplete with that 11 amino acid.
Right, make the incomplete protein with the 11 amino acid. The thing is that this breakage of your messenger RNA could happen in any messenger RNA in any place. So this is a now specific mechanism for dealing with lots of non-specific problems that could happen. And so, the eat me signal at this end says, eat this part, but also eat all the rest of this too, and has no idea what it is. So, which might be dangerous if you have kids who eat this, but everything else which is there, good for vegetables. No.
So then is the broken mRNA, since it gets dissociated completely from the ribosome, then that's degraded by the similar mechanism? Or?
Yeah, so the question is what happens to that messenger RNA which is lost? That gets degraded by the exosome. You remember the exosome with all these extra exonucleases which will break things down? Okay, so that's what happens if you have a stalled ribosome in a bacterial case. And it turns out tmRNA just works in bacteria. There's also a case that can happen in eukaryotic systems. And this is what's called nonsense mediated decay. So what the heck is nonsense? Nonsense codons are the stop codons. So if now you have a stop codon which happens too early in your messenger RNA, the ribosome will also stop here. And in the case of a eukaryotic ribosome, it's reading along a messenger RNA which has been spliced. Now, what happens when you have splicing? Splicing then will introduce these exon junction complexes wherever you have the two exons that have come together. Normally, these exon junction complexes get removed as you have translation, and then the ribosome will get to this particular stop codon. If, however, you get to a stop codon and there's still one of these exon junction complexes, which is here, this means, this stop codon happened too soon, because it should be over here after you've done all the splicing. And so because of that premature stop codon, now you have signaling that does happen through the ribosome, which says, hey, exon junction complex, some of these proteins which are here, time to turn on a enzyme which will get rid of this cap. Once you remove the cap, now we've got lots of exonucleases which will chew in at various ends. These are endo and exonucleases. It's just shown here as an endonuclease, but I like to think of this as an exonuclease, which chews up your messenger RNA. Why is it important that it chews up the messenger RNA? Because this messenger RNA clearly has a stop which has happened too soon because of the exon junction complexes which are still associated with the RNA. So that's one issue which can happen. The other issue is, well, instead of having one too many stop codons, you lose your stop codon. What happens if you lose a stop codon? Well, if you lose a stop codon, then you have non-stop mediated decay. So what's at the end of the vast majority of eukaryotic messenger RNAs? Poly A tail. If you keep translating through a poly A tail, what are you going to do? You're going to translate all of those AAA residues. We haven't learned the genetic code, but AAA stands for lysine. So your protein now has a whole bunch of lysines at the end of the protein, which serves just like the eat me signal that you have from tmRNA. Anytime you've got a whole bunch of lysines at the end, this now serves as a degradation signal for the protein, but also the ribosome is going to get stalled here at the very end of that messenger RNA. It needs to get degraded as well, and so there are other proteins which will bind here, including the exosome, which loves to chew in at your messenger RNA, and this ribosome is also now released. You can go back and restart that whole translation process. So, any questions on this? Yes. Yeah.
Are you signaling, like the degradation signaling messages related to like the ubiquitination pathway, or is it just proteins instead of?
Okay, so the question is, are these degradation pathways linked to ubiquitination? And the answer is no. These are separate degradation pathways, and we're not going to get to ubiquitination degradation today. But yeah, they're completely separate, and they do seem to be really this whole quality control mechanism. It's getting rid of proteins, like yeah, with the tmRNA and with the extra lysines at the end. These are clearly incorrect proteins, and so those are going to get degraded. Yeah.
The exon
Junction complex?
yes.
Mm-hmm.
The exon junction. How does the ribosome recognize them separately from any other codon?
Okay, so the exon junction complexes are proteins which bind to the exons and where the exons have joined to each other. And so it's the messenger RNA. So again, when you look at these animations, you've got the nice yellow strip, and that's all that it is. It's just the messenger RNA. No, it's messenger RNA plus protein. And so those proteins then are part of the complex, this messenger RNA complex, which then can talk to the ribosome and give you their signaling. Yeah, Olivia?
So the nonsense mediated decay, non-stop mediated decay are only in eukaryotes.
Yes, so nonstop and nonsense mediated decay are only present in eukaryotic cells, and tmRNA is only present in bacterial cells. Okay, now we ready for our clicker question? Yes? Great, because it leads right into this. So, the presence of which of the following on messenger RNA leads to nonsense mediated decay? Cap binding complex, EF4E, EF4G, exon junction complexes, and EF2. Let's get 100%. Let's go for it. Vote early, vote often. And I won't make any political commentary, even though I'd really like to. 7, 5. Okay, do we get 100%? No, not quite. Very close. Yes, it's the exon junction complexes. It's the whole nonsense. And nonsense means, again, that you've got a stop codon which comes too soon, because that's the nonsense codons. Okay, so we've gone through all of our translation. We have our polypeptide, which can fold up into a protein, assuming it doesn't have all these eat me signals and getting degraded, et cetera. Did want to mention one other thing, just as we're moving through here, is because, again, translation is one of the most energetically important things that happens in molecular biology of a cell. It's also the target for a lot of antibiotics. And as we've talked about today, there are a certain number of differences that you have between bacterial ribosomes and eukaryotic ribosomes. And fungi have figured out how to do this, and some bacteria as well, over millions of years of evolution. And then we've figured out that we can take some of the chemicals that these fungi and other bacteria produce in order to block the growth of those particular organisms, and usually it's going to be ribosomes. So all I did here was take our table of the various different antibiotics and just circle all the ribosomes, and hopefully it's really obvious that many of these commonly used antibiotics are polypeptide ribosome, 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 ribosome. So many antibiotics, not all of them, but many antibiotics are targeting the ribosome because the ribosomes are different enough between bacteria and eukarya, i.e. us, that we can use antibiotics to treat bacteria and particularly bacterial infections. So we've made our polypeptide, but that's just a stretch of amino acids. In order to get function, we need structure. Exactly. So how do we get structure? Structure in proteins is due to protein folding that we've talked about before. We have a polypeptide, usually coming off of the ribosome, and then it will eventually fold into its final structure. Sometimes it'll bind to some kind of cofactor. Sometimes there'll be modification. Sometimes you have other subunits that are associated. But this final structure is mostly, and we talked about some of the exceptions before, amyloids and structures that are, say, proteins that don't have always the same kind of structure. But for the vast majority of the time, one particular amino acid sequence is going to give you one particular structure. How does this happen? So it turns out that the ways that proteins fold are actually really similar to the way we've already talked about them. You make primary structure, then you have secondary structure, which forms tertiary structure. And so that secondary structure is the first thing that forms. And many proteins go through this process called the molten globule process. And all that a molten globule is it's your protein that has most of the secondary structure elements that are normally associated with a protein and a little bit of tertiary interactions. And so this is a classic example of one of these molten globules. You'll see here it's got all these helices, but this one is a little bit too short, and here's our final protein. So how do you go from starting this process to actually finishing it? Here's our ribosome making our protein. You end up getting some of these secondary structures. Sometimes you'll have all of the completed folding that happens here. This is actually a rarity. Most proteins don't actually seem to be able to fold completely as they're coming off the ribosome. Most of the time, they go through these molten globule steps, and they need chaperones. What the heck is a chaperone? Again, as a dad, I really like this analogy. The chaperone is something which makes sure that the wrong interactions don't take place. So with my daughters, I have a chaperone to make sure wrong interactions don't take place. So that's really what this is about. You don't want the wrong interactions taking place. And the wrong interactions mean the parts of your protein coming together incorrectly and giving you a non-correctly folded protein. So we'll stop here. I'll continue to talk about some of these chaperones, how they actually work after the exam. So Wednesday will be review, Friday's the exam.